Good morning, everyone. I would like to thank Jill Morgan and the OncoAlert Network for the opportunity to present today the results of our analysis entitled Prognostic Implications of Risk Definitions from the monarchE and NATALEE trials, recently published in the Journal of the National Cancer Institute. As we know, the combination of CDK4/6 inhibitors with adjuvant endocrine therapy has improved the outcomes in patients with hormone receptor-positive HER2-negative disease at high risk of recurrence. Both the monarchE and NATALEE trials demonstrated improved outcomes in terms of invasive disease-free survival and also a significant reduction in distant recurrences, leading FDA and EMA approval to the use of abemaciclib and ribociclib in the adjuvant setting of hormone receptor-positive early breast cancer. However, since there is no standardized definition of disease at high risk of recurrence, monarchE and NATALEE trials apply different inclusion criteria. The monarchE trial criteria were more selective, including only patients with node-positive disease, while the NATALEE trial inclusion criteria were broader, including also patients with node-negative disease. Therefore, only part of the two populations included in the trials overlaps, and a better understanding of the long-term prognosis based on the inclusion criteria of the two trials could be crucial for selecting the optimal adjuvant endocrine therapy in this population. In our analysis, we evaluated individual patient-level data for women diagnosed with hormone receptor-positive HER2-negative early breast cancer, including in the 3 large adjuvant phase 3 clinical trials conducted in Italy. The main objective of our analysis was to assess the prognosis of these patients according to the inclusion criteria of the monarchE and NATALEE trial. Among 4,795 patients included, 28 were classified as high risk according to the monarchE trial, while 56% were considered high risk according to the NATALEE trial. Patients were then divided into 3 different cohorts: the concordant low-risk cohort, meaning patients at low risk for both monarchE and NATALEE trials; the concordant high-risk cohort, meaning patients at high risk for both the monarchE and NATALEE trials; and the discordant risk cohort, meaning those patients had high risk for only one of the two trials. Among the patients included in our analysis, 28% were classified as high risk by both trials included in the concordant high-risk cohort. 43 patients has been included as concordant low risk by both trials included in the concordant low risk cohort, while other 28% of the patients were classified as high risk by NATALEE trial only and included in the discordant risk cohort. At a median follow-up of 7 years, the 7-year disease-free survival was 69% in the concordant high-risk cohort, compared with 89% in the concordant low-risk cohort, and 84% in the discordant cohort. But the main finding is reflected in the 7-year overall survival, which was 96% in the concordant low-risk cohort versus 95% in the discordant cohort, statistically significant with an hazard ratio of 1.52. While the 7-year overall survival was 84% in the concordant high-risk cohort, with a statistically significant difference with an hazard ratio of 3.38. Therefore, our results indicate that patients deemed high risk per NATALEE trial only seems to have a 7-year overall survival only with 1% lower than concordant low risk, while those patients at high risk per monarchE criteria showed an important poor prognosis. While waiting for more mature follow-up data from the monarchE and NATALEE trials, particularly with results in terms of overall survival, our results may help physicians and patients to perform more informed discussion and the need to escalate the adjuvant endocrine therapy based on their actual baseline clinical risk of recurrence. And I would like to thank you all for your attention.